Hello, my name is Madeline Winans, and I am a Chair Master's student working with Dr. Kelly George. I'm going to be discussing part of my master's research project titled Measuring the Effect of Facility Relocation on a Population of California Sea Lions' Welfare. Columbus Zoo and Aquarium has recently built a brand new region that is home to California sea lions. In the early stages of this process, Columbus Zoo and CHAIRE identified the need to monitor the welfare of these animals throughout this long-term transition process, and that's where our project comes in. The overarching goal of this project was to assess potential changes in welfare in response to relocation from a temporary facility out of state to a permanent habitat at Columbus Zoo. The objective of the phase presented here was to establish baseline welfare measures for the animals prior to relocation. So meet our study population. The group consisted of 7 animals, males and females, and 1 pup was born during the study. 2 other parameters of the study were period and staff, where staff 1 indicates a variable staff group and staff 2 was a consistent staff group. We collected data over 17 months, including both behavior observations and hair samples to measure cortisol concentrations as an indicator of potential chronic stress. Hair cortisol has not been measured in this species before, so this was a novel approach to assessing their welfare. Cortisol is a hormone released as part of the physiological stress response and can be measured in blood, saliva, feces and urine, and hair. We chose to measure hair cortisol as it reflects long-term cortisol production, and the sampling procedure is trainable and minimally invasive. For our methods, the sea lions were trained with positive reinforcement to participate in the hair sampling, where the trainer shaves the same patch above their rear flippers at each sampling time. To record behavior, we utilized scan sampling in 10-minute intervals and a species-specific ethogram. We hypothesized that sex would have no effect on either parameter, hair cortisol would be negatively correlated with age, period, and staff change, and inactive behavior would be more likely than active with increased age, later periods, and staff group 2. We analyzed behavior with odds ratios, or comparing the likelihood of the animal performing inactive behavior over active behavior. An odds ratio greater than 1 means inactive behavior is more likely, and less than 1 means inactive behavior is less likely than active. We found that inactive behavior was more likely during periods 2 and 4 compared to period 5, and less likely in period 1 compared to period 5. Additionally, inactive behavior was less likely than active where there was a variable staff group compared to the consistent staff group. This figure shows the mean hair cortisol in each period, with staff group 1 in gray and staff group 2 in blue. A main finding here was that mean hair cortisol concentrations were significantly higher with staff group 1, the variable group, compared to staff group 2, the consistent group. We see a significantly elevated mean hair cortisol in period 2, which is partially due to the birth of a juvenile which occurred during this period and whose hair cortisol values were much higher than the adults. This is consistent with findings regarding hair cortisol concentrations of newborns in other species. Even without the juvenile, we still see the same significant difference between staff groups, and mean hair cortisol in period 2 is still significantly elevated. This may be due to the occurrence of breeding season during this period, which has been shown to impact cortisol concentrations in other species. We found that sex had no effect on either parameter, which may be due to the skewed sex ratio in our sample. Also, age had no effect on behavior, but younger animals had significantly higher cortisol concentrations, which is consistent with findings in Hawaiian monk seals. So one of our key findings was that change in staff significantly affected hair cortisol, which is supported by literature showing that zoo animals respond differently to familiar and unfamiliar people, and unfamiliar staff has been linked to increased hair cortisol concentrations. Even with high variation, our population's overall mean hair cortisol concentration does align with hair cortisol values in related species. This value will be recalculated with data from the acclimation period to obtain a more representative range for California sea lions. So the next steps for this project include analyzing the data collected during transportation of the animals, as well as continuing to measure both behavior and hair cortisol while the animals are acclimating to their new environment at Columbus Zoo, both to measure the effect of facility relocation on the animals' welfare. Additionally, there are 2 other related projects underway. First, we are assessing Columbus Zoo visitor attitudes toward our study species and their welfare in zoos, as well as conducting an experiment to determine the degree to which the species can see in color, an unknown biological trait of the species. Overall, we hope this project will be beneficial in offering evidence of a novel approach to sea lion welfare assessment, expanding our knowledge of the species, and contributing to the growing field of marine mammal welfare science, particularly for zoo-housed pinnipeds. On behalf of our entire research team, thank you for watching this virtual presentation. If you have any questions, please direct them to either of these email addresses. Thank you.